I'd like to pitch this lecture in a different sort of way. Imagine, whether you're a professor or a student, in either case, you have a pet project and you're very enthusiastic about it, very protective maybe, but also obsessed. Now imagine if you could write a proposal that could recruit 7 or 8 other faculty experts to help you with that project. Do you want to keep it all to yourself, or are there barriers that you're not disciplinarily prepared for? Well, that's my problem. And so I had a chance to write such a proposal to the National Science Foundation. I found 7 to 8 other faculty members that were very enthusiastic about it. And then, to my utter disbelief, it was funded, and the funding is for 7 years, and we can support as many as 15 graduate students or postdocs. So this is a real treat for me, and it's made my project, which was always viewed as somewhat arcane and eclectic, into something that's become more mainstream, which doesn't matter to me. Mainstream isn't always good, but it's really advanced the science. And the science has to do with how sea creatures attach to surfaces underwater. So what I'd like to do is progress through this proposal and then show you highlights of progress that have occurred in this team. What we had to do in the proposal is to pitch a grand challenge, which is to understand the design principles of wet adhesion. What wet adhesion means that makes it different from just taking a bit of super glue and sticking your fingers together is adhesion under hostile conditions, which includes moisture, of course, but also salt, corroded surfaces, biofilms, and very substantial hydration layers that have to be pushed aside in order for adhesion to occur. And we have 2 model systems for this, the mussel and the sandcastle worm. They use very similar chemistry, but this system is more complicated because it's actually got to make an interface with these extra-organismic fibers that have no living cells in them to living tissue on the inside. So they're a bit like an implant, a bionic implant. And this one is just sticking little sand grains together like a brick mason. So it only has to deal with these interfaces between the glue and the inert particles that it picks up from the intertidal. So to understand how adhesion works in these systems, enough to be able to translate it into synthetic systems, and ultimately to pioneer a systems approach to using properly engineered polymers in a systems approach that is at multiple length scales. The faculty are shown here, and I just project this to show the range of specialties of the disciplines. And we have 2 affiliates. Many of you will know Matt Terrell. He's kind of a leader, and he was the dean at UCSB at that time, so he got involved. But he's still collaborating with some of the team members. And this is a very important slide that shows that our team is not artificial for the purpose of selling a proposal. These nodes, so to speak, that mark these intersections between faculty members indicate the number, or at least more than 2 publications that have resulted during the collaborative research. It's a beehive of activity, as it says, and I've interacted with most of these, with the exception of the theorist here, who's waiting for his turn. You know, they need to have everything precisely defined before they want to start to model things. But he's doing it with simpler systems made by Craig Hawker's group. These are the students and postdoctoral trainees that, as of last year, there are many that have graduated already, and quite a few of them work in my lab. And this is the process that we're hoping to take in the research, which we call discovery to translation. Muscle adhesion depends on things that happen at different length scales, from the chemistry at the interface to the architecture that's supramolecular, to the geometric shape of the plaques used in adhesion, to the whole animal, and ultimately even to a higher length scale that involves clusters of these animals. You have zebra mussel problems, you know they don't occur as individuals, they occur and cause fouling problems by the clusters that they form. So that would be an example of a living composite material where these threads are the continuous matrix and the shells of the animals are the inclusions. So some of us focus primarily on characterizing things at different length scales. I do a lot of biochemistry at the nanometer scale. Others look at architecture and geometry effects, and they feed their models or their sequences to the second group in the team that uses their expertise, for example, in surface physics, to measure the adhesion that's possible for different proteins in the complex solution that's deposited by mussels, to look at their viscosities, hydration, etc. And that sets up a structure properties database that is then accumulated and drawn upon by the members of the team that are interested in synthesis theory or simulation. They'll make synthetic simplified versions of some of these polymers that will lend themselves to modeling and to more tests of surface physics, etc. So there's a lot of resonance between these groups, and progress depends on the distillation of essentials by these two groups and the translation to synthetics by this group. So the way I've set up this seminar is to talk about progress on these mussel or sand kesselworm derived adhesives according to the 4 universally accepted requirements for good adhesion. Every adhesive, practical adhesive, needs to fulfill these 4 requirements, which include strong interfacial interactions on wet surfaces, despite the high water dielectric, etc., etc. Removal of weak boundary layers. That actually precedes this one. The deposition of a dense water-insoluble surface-active adhesive liquid. Now, you see, when you buy your 2-part epoxy from the hardware, you're just squirting it out onto a dry surface. Hopefully you've cleaned it off with something before you put it on there. But these organisms that live in water deal with a weak boundary layer all the time. It's part of their system. And so when they come up with their precursors, they can't diffuse into the medium, or the game is lost at time 0. So they have to be phase separated from solution. And finally, this precursor, adhesive precursor, has to cure and toughen underwater. So we can look at those basics, taking one at a time and just pointing out highlights from the mussel and how those highlights have been translated. If we start with strong interfacial interactions, I need to zoom on a cluster of these plaques and show one in cross-section schematically to show you the distribution of many different proteins. Some are really dedicated to working at the interface between the plaque and some foreign surface. And we do have evidence to suggest that it isn't the same cocktail, that to some extent the mussel can sense certain properties of the surface and create a cocktail that's tailored to those properties. But if we just focus on this one interfacial protein, MFP5, stands for muscle foot protein 5, and look at its sequence. This took many years to do, but now that we have the sequence, it's part of the structure properties database. And what stands out about this protein is that all of the tyrosines, the Ys, which are monohydroxy compounds, have been functionalized with another hydroxy to make this a catechol functionalized protein. And the catechol content is 30 mol%. That's higher than any other amino acid. But there are also biases in other amino acids. The positively charged ones are definitely always there. And if you add all of them up, including the Ks, the Hs, and the Rs, it comes close to nearly 30 mol%. The glycine levels are high, and there's a variable, there's a wild card here as well, in that there are serines that are phosphorylated variably. So those residues are not like tyrosine that are universally converted to DOPA. That probably, the phosphorylation, has something to do with the mussels detecting something on the surface and then deciding whether or not to put phosphate in. Phosphate is great. Do you know this stuff we get in the U.S. hardware stores called navel jelly is phosphoric acid or a polyphosphate that stops corrosion dead, and then you can paint over it. So that kind of chemistry has been well known as an anti-corrosive for a long time. Okay, so the catechol functionality is very important, but we can test how important it is using the surface forces apparatus, which is shown in very general schematic here. It basically involves two hemispheric surfaces brought together, round side, that have mica, atomically smooth mica, glued over them and silvered on the back sides. The protein is applied as a monolayer to one surface, and then it's brought together in the approach. There's the hard wall, which means you can't compress it any farther. And for proteins, actually, that's a useful measure of the radius of hydration, the diameter of hydration. And now you can see from these traces that the adhesion for this protein is very fast, where at 5 minutes it reaches 80% of the maximum adhesion. And this adhesion, this maximum adhesion that's been observed, is about, in energy terms, using Deryagin approximation, is 14 millijoules per meter squared, which is higher than the strongest known non-covalent biochemical interaction between streptavidin and biotin. So it's strong, all right, and it almost suggests that in its evolution of an adhesive, the mussel has gotten very familiar with aluminosilicates and what they need in terms of an interaction with an organic for good sticking. All right, so that looked very good, but then when we looked at the effect of pH on adhesion, the loss in adhesion was almost absolute by going from 3 to 7.5. The pH of seawater is pH 8. So this is for MFP3, again, in asymmetric configuration. For 5, if we just look at this peak as a function of pH, we have more points. It goes down. And one has to ask, since this is biochemistry or started as such, why an animal in nature would do such a thing. To live at pH 8 but make an adhesive that failed at pH 8. And the best explanation for what happens in this pH-dependent adhesion is that these dopa residues spontaneously oxidize to quinone. If the adhesion is based on the bidentate hydrogen bonding or coordination of oxides in the mica surface, then the oxidation to quinone would abolish the adhesion, because this here depends on a hydrogen bond donating ability, and you don't have any donating ability in the quinone. So the fact that the dopa residues are prone to oxidation at pH 8 only means that the mussel has to protect what it's doing so that this spontaneous autooxidation doesn't occur. And I can tell you furthermore that once this catechol is engaged in a bidentate hydrogen bonding mode to the surface, it is no longer prone to oxidation. So the mussel has to give these proteins enough time to go to this state before it exposes the system to the quinone. Let's see if this is biologically relevant. And this is a kind of study we have to do in order to translate what's actually happening in the mussel to a useful synthetic system. To test what the mussel is doing with its foot during the formation of a new thread, which takes 5 minutes in an adult mussel and 30 seconds in a juvenile, we prepared a mica surface that has covalently tethered to it a bilipid membrane, and attached to that, the amino groups on the bilipid on that face is an Oregon green pH sensor, fluorescent pH sensor, that has a pK of 4.7. And now we put that on the surface and expose it to several cycles of seawater to make sure that we get a decent titration curve through the different pHs. And after an initial curve, so there's a hysteresis after the initial curve and all subsequent curves. So the slides have to be conditioned with an initial exposure, and then we can present that to the mussel, and using confocal microscope can measure the pH under the tip of this foot while a new plaque is being made. That shows the pH under the foot to be like our stomach pH. It's pumping protons in there along with the adhesive proteins, and they have about this much time to stick to the surface before the foot lifts off and the pH equilibrates to the seawater. Now, are we constrained by this particular process? One would hope not, because if you're going to repair something in the body, let's say on a bone or an implant to a bone, you don't want to have to squirt a lot of acid in there. So is this a technical constraint? And a student in my lab, a group of students in my lab showed that it's not a constraint if you use borate buffer, in that borate at pH 7 to 9 forms these stable complexes that are not more stable than the bidentate hydrogen bonding on the mica. So there's a lag time for this exchange, but it is favorable and it does happen, and the adhesion strengths that are measured are comparable to the adhesion that's set under the acidic pH. So this would be a possible procedural technology for adhesion that had to be done at pH 7 without losses to oxidation. All right, so I can tell you about a couple of translational projects that combine the the concept of lots of catechols for adhesion with the acidity of the environment. This has to do with a project that was called pillared flypaper, in which the investigators used etching, stamping, lithography to create these silica pillars that were coated with PDMS that was functionalized with eugenol that was initially blocked. So eugenol is the precursor for DOPA here. It lends itself to click chemistry. And then in a last step, after clicking them on, the triethyl silyl groups can be removed by acid treatment. So that's called the deblocking. And if you use AFM to measure the strength of adhesion of these glass beads that were in a pan initially, and the pillared shape was pressed into the pan, and the beads stuck to these PDMS modified tips, that you can create polymers with different catechol density by the feed in this polymer synthesis, and then you can measure the correlation of adhesion to the concentration of catechol in that polymer, and it looks roughly linear, and it goes up to 20%, which is almost as high as it goes in the mussel. So that's one. This is all wet adhesion, and it's carried out at acidic pH, and once the beads stick, you can change the buffer of the system to a higher pH, and the performance of adhesion is still good. But if you raise the pH before you push the pillars into the pan of beads, you lose your adhesion to silica. So that's the processing importance. Another application is this self-healing paper, in which Colby-Ahn here made polyacrylates that were functionalized again with the eugenol by click chemistry, and these could be either blocked or deblocked and tested for healing of amputation here. I guess you could call this a polymer amputation. You use a razor blade. You can do that underwater, or you can do it dry and then put the severed pieces into water. If you've deblocked these groups, slight pressure causes these two cut ends to heal spontaneously. And if these are not deblocked, there is no healing. And the interesting thing is, if you pull these again under tension, the breakage never occurs at the heal site. So the healing is stronger than the cohesion of the polymer initially. And I don't know what the structure is across the interface. It's probably a very dynamic thing, but we are guessing that there's some kind of bidentate hydrogen bonding that initializes the healing process. All right, so let's go on to question number 2, removal of weak boundary layers. It's intuitive to everybody that if I squirt water onto this laminate desktop and then try and stick a piece of Scotch tape to the laminate, it won't stick until I remove the water. But actually, you can get some sticking if you push down really hard on the tape. And maybe your physical force is dehydrating some of the surface. But if you take away the force, the water will tend to wick back into that interface. So by sticking to wet surfaces, these organisms are doing something quite special. This is another one of the interfacial proteins. Again, it has a lot of mole percent DOPA, and it has to get through this hydration layer. Not to mention, it has to get through the bulk, but usually, well, you'll see how it handles that. How does it get through that hydration layer? One of the ways in which we've investigated this, particularly a postdoc in my group, Waite, has used the ATR, Fourier transform infrared spectrometer with a flow cell to apply fairly uniform nanobead materials like TiO2 and hydroxyapatite to increase the surface area in that sensor cell. A solution is flowed through it with protein in it. That would be this MFP3 protein, and the infrared then by time-dependent changes in intensity at different wave numbers informs on what's adsorbed or, in the case of water, what's desorbed. So I only want to focus on this part of the spectrum, which has specific absorbances for chemistry like bending of CH2 and stretching of NH. But these are very sharp peaks in the spectrum. The broad tendency of the peak is related to the bending and stretching modes of OH and water. So these are just, these finer things are punctuated on top of that. So in the MFP3 variant that was synthesized by solid state synthesis, you see actually a buildup of hydration with some of the other chemical signatures. But in the version with DOPA, you have negative absorption, indicating displaced water, according to the interpretation of these spectra. So it doesn't tell us anything about the mechanism. We know that these catechol are capable of bidentate hydrogen bonding to the surface, but how do they displace the water? Another technique that we've used to look at hydration is called Overhauser dynamic nuclear polarization. And this is a method that's been pioneered by Song-Yi Han, who's the spectrometry expert in the group. And it's based fundamentally on labeling a surface covalently with a spin label. And that's what these little yellow beads are. The spin label has a magnetic moment, and it can be perturbed by electron spin resonance. And if a water molecule with two protons or hydrogens on it is within 15 angstroms of it at a so-called Larmor frequency, there is an Overhauser effect from the electron in the spin label to the proton that can be detected in NMR as a polarization with a nice sharp peak that then decays in time. And the decay is an indication of how rigid or dynamic the water is within that 15 angstrom diameter. So she can apply this method, spin label dependent method, to a number of proteins to test how they change water structure in the vicinity of the spin label. And many of them here, so this is just the polystyrene beads, control labeled with a spin label. Polylysine, PEG, and different proteins recombinant or native with different amounts of DOPA. And these data are telling us that on the polystyrene surface, the DOPA doesn't play any role in dehydrating the surface. What does play a role, though, with this precursor MFP3F, which doesn't actually have very much DOPA in it, is the very high level of tryptophan. She's visualizing a kind of pi-pi or aromatic hydrophobic interaction with a surface that helps drive the water away. So far, this result really can only be applied to hydrophobic polymer surfaces, and mussels have only seen those very recently in their evolution. But their approach seems to be based on hydrophobic content. So I've spoken a lot about DOPA and other aromatic hydrophobic residues. What about others, particularly the positive charges here in this sequence? Dissecting the role they play in adhesion is very difficult with a full-length protein. So the synthetic coordination chemist in our group decided to pick one siderophore. Siderophores are low molecular weight, frequently catechol-containing molecules that bacteria use to sequester iron from their environment. Iron is almost always limiting in natural environments, and they need it for metabolism and growth, etc. And one that she found, which is not marine, it's actually from a, this animation isn't working. It's a tricyclic lactone. It has a lysine and a catechol in 3 identical environments in this siderophore. So the catechols and the lysines are in identical environments, which gets around the problem of this, the environment is sequence dependent, all right? So they made a synthetic version of this to make large quantities and basically replaced the lactone ring system with this tren shown here. The tris-aminoethylamine, and then the catechol and lysine continued in each of these R groups. And these were tested for adhesion to mica, and when they had both the lysine and the catechol ring present, they showed great adhesion. When they had only the amine present, but some shortened form of the dihydroxy, the adhesion was reduced by at least 90%, usually more. And when they had the catechol present, but the amine was blocked by acetylation, it was also at control levels, buffer levels. So this is telling us that the catechol works on the surface, at least in salt solution, only when there's a cation flanking it. And we can also look at the effects that this has on hard wall. Remember, the hard wall is an indication of the hydration diameter of the protein, but in this case, we're using the same peptide construct, and the hard wall is decreasing as we go to the fully cation and catechol functionalized versions. And that was interpreted after much analysis to indicate that the lysine is helping to evict a hydrated cation that's adsorbed to the surface, which is going to be a real problem in seawater because there's so many cations floating around. To model this, to animate this slightly for your understanding, well, first of all, let me tell you that in mica, which is always selected for SFA because it's atomically smooth, when you make a cleavage plane, it has potassium sitting there on the surface. If you put the mica in distilled water, usually that potassium will come off. But if you run it in salt, which these tests were done in, potassium nitrate, the potassium is going to be replaced. And so that's sitting there, and the catechols don't know what to do about it on their own. So what happens is that this construct, which is first floating around in solution, has to get a toehold by kicking out a hydrated potassium ion, and it's only after that's done that the catechol can bidentate hydrogen bond the oxide's underlying oxide surface. So this is fairly plausible, but it needs to be tested on other surfaces, and certainly needs to be modeled, in this case, on some well-characterized surface. So getting around to the third requirement, we need a dense, water-insoluble, surface-active, adhesive liquid. And this was first modeled on the basis of what sandcastle worms did. They used polycations and polyanions, mix them together to form this thing called a coacervate, and they deposit that onto the sand grain. Let's look at the history of this thing called a coacervate. It actually goes back to just a year after my birth by this Dutchman here. And he described a process by which you take, and he took mostly biologically derived polyanions and polycations. Polycation here, polycation, polyanion with their microions. You put them in solution, you adjust the pH so they have exactly the same countercharge. They neutralize each other, and then when they neutralize each other, instead of precipitating out of solution, they phase separate as a fluid, a separate fluid that's rich in polymer, partially dehydrated, but not completely, and still fluidic, and you can actually centrifuge that dense phase and harvest it. So it's called a coacervate, and it's complex because it involves two complex polyions to start. These have wonderful and still somewhat mysterious properties. Low interfacial energies, generally lower than 1 millijoule per meter squared, so almost anything that you put in solution will be surrounded by this complex fluid, engulfed, they say. The concentrated polyelectrolytes are denser, yes, obviously. Molecules still have very high diffusion coefficients in the coacervate, even though the polymers become more concentrated. And they have very peculiar viscosity behavior where the viscosity of the coacervate is less than 1/10 that of either polyelectrolyte at the same concentration. None of these would be predictable. So the synthetic group and the synthetic teams in our group have made coacervates from tri-block copolymers, where one is positively charged at the ends, the other negatively. Usually they use guanylyl and sulfonyl groups. And when they mix them together, they form these gels that have these little nodes in them where these oppositely charged tails are concentrated. So they're like physical crosslinks holding this hydrogel together. And if you increase the salt concentration, you can disperse that. And they've actually started a company in the San Jose area that makes this tri-block as a hair conditioner. Works quite well, they say, though I'm not one to test it. All right, so that's an application. NSF is always happy to see those. Well, let's get back to the Mussel holdfast here and the proteins it contains. One of the proteins that covers the whole structure is called MFP1, and it's shown here. It's got a repetitive sequence that's repeated up to 75 times in the native protein, and it can be coacervated with hyaluronic acid, which is a very popular building block for biomedical materials. And when those two are coacervated with each other, the diffusion coefficient of water in the two is exactly the same. So they have to have some way of getting around in that water for that to happen. This is a cryo-EM of a TEM of a coacervate showing that it has structure. And I'll move on to this, which is a property that's rarely talked about with respect to coacervates, and that is that these coacervates with hyaluronic acid were tested somewhat serendipitously in the surface forces apparatus for their behavior under friction. So you bring these two faces together, mica on mica, and then you do this at different loads, and then you can look at the damage that occurs on the mica surface in a load-dependent fashion. And it turns out that coacervates prevent any damage from occurring to mica, even with the highest load cell. So this goes off the top. If the mixing ratios of the two are less than optimized, then you get damage. So it has something to do with damage. And these two tests with recombinant protein show that DOPA has a role in protecting the mica surface from damage and possibly by lifting the slip barrier. So if DOPA binds very tightly to the mica, then the slippage is not going to occur at the mica. It's going to occur higher up. This needs to be further explored. All right, so the invention by the Mussels in this business is that they figured out how to make coacervates with only one protein, not two. Those are called simple coacervates, even though in most respects they're more complicated. And again, it's pH dependent. This is coacervate yield according to turbidity. And the coacervation of this protein is very important to its adsorption by QCM to this hydroxyapatite surface. See, the blue, which is the optimized coacervate, has the highest adsorption. And the last category, how to cure and toughen adhesives underwater. I'll just mention a few. This one in particular because I mentioned it to Boschin students. The thread has a cuticle on it that's thought to be coacervated, and this is the protein, the only one of the pair of proteins that's been well characterized. It is bound to iron. It binds iron very well, and if you take these sections of this cuticle and do resonance Raman on it, you get this kind of color enhanced image of those portions that show fingerprint resonances for iron catechol complexes. So the complexes are enriched in the cuticle and fairly lacking in the core, and that means that these proteins, at least in the cuticle, are cohesively stabilized by these metal interactions. That was mimicked by a synthetically inclined student in my group who replaced the catechol with something less prone to oxidation, this mimosine-derived dihydroxypyridine. And he actually made hydrogels with different metals, aluminum, iron, or indium, and did dynamic tests on them. And if you know your dynamic tests, you know that the intersection of the G prime with the G double prime is proportional to 1 over the bond lifetime, or tau. Although each of these complexes has the same stability, they have different lifetimes that can be used to make hydrogels with different properties. So tunability, basically. And the last thing that I really like about this project, because it's graduated us from a focus on the nanomolecular aspects to higher length scales, is this business of testing the plaques directly with a really good and versatile tensiometer that has microscopic inspection, it has angle adjustment, it has rate adjustments, and these kinds of force extension maps can be turned into fracture energy. On these plaques, and we've done hundreds of them, the fracture energy is averaging 100 joules per meter squared. And if you recall, our stickiest protein in the surface forces apparatus was about 14 millijoules per meter squared. So heroic behavior at the interfacial level is much, much lower than heroic behavior at higher architectural levels. And that's because of energy dissipation. How does energy get dissipated when you just load on more protein? Possibly by all this porosity. Pores are good at stopping cracks. We don't know, but that's one direction we're going. Also by geometry, the spatulate shape adds a lot to increase the force to failure. And this one effort that was done by Zhang Zhao in my group to make something, a higher length scale, porous material, along the lines of what the Mussel does, involves one polymer that's catechol functionalized, it's polyacrylic, and another one that's a chitin that's functionalized with these trialkylamines. They can be dissolved in DMSO if you add a counterion to that amine, because the polyacrylate will not be charged in DMSO. And then when you put a drop of this on a glass slide in water, DMSO comes out, water goes in, there's a phase inversion, suddenly turns into a foam that's solid, that sticks to the glass surface underwater. So I think that's got a lot of promise, and it's a paper submitted. Summary, these are the accomplishments. Some of them are substantial, some are little steps, and we have a lot of work to do, but we still have 2 years to go on the grant, and I thank you for your attention. In solution, they are really lacking any secondary structure. But that doesn't say they don't acquire it in adsorbing to the surface. That's just a little harder to measure. We can do it by FTIR, but I have less confidence in it than circular dichroism. But the proteins don't have structure by circular dichroism. But yeah, also, you look at them. The charge density is so high, and it's all the same. They'd just be repelling themselves. Yeah. Yeah.
So you one slide you say that the amine, right?
Yeah.
So that the catechol is more for kicking out the potassium. Right?
Yeah.
So what if you do this just in like water without all these salts?
Yes.
Right?
Then you get adhesion.
Then if you add the salt back, it's
Yeah.
still strong.
If you adsorb it under distilled water conditions and add the salt back, I believe they did that and it's still stuck. Yeah. So the ions won't go in and push the catechol out. Right.
Effect, right?
Yeah. Right. But I don't believe that anything in biochemistry is monofunctional. So, for example, I can make the argument right now that if coacervation is important in how these proteins work underwater, you need the positive charges for coacervation. Viral origin. You mean so that you could use something like a phage display to select? Yeah, I haven't done a lot of database searching along those lines, but it wouldn't be surprising, because actually lysine, arginine, histidine proteins that are intrinsically unstructured is another way of saying histones. And everything has histones. I'm sure there are. There's a very good group in Singapore associated with a former postdoc of mine, Ali Miserez, and they're looking at adhesion by the green mussel, which is strictly tropical. And the sequences are different. The proportions of residues are slightly different. But one thing comes to mind in answering that question. If you look at the adhesive proteins of the zebra mussel, and I've sequenced a couple of those, and Eli Sohn at the University of Toronto has done some more. They don't have catechols flanked by lysine. So that might suggest that in being specialized to fresh water or low salinity brackish water, they didn't have the challenge of having to replace hydrated cations, right? So that would be interesting to look at, to see if they actually lose their adhesion if they're tested under seawater conditions or less than.
Just pick a zebra mussel, throw it into the...
Well, no, you couldn't do that. I think it would kill them. But if you isolated the protein, you could test it in the surface forces apparatus or AFM under high salinity conditions. Yeah. Yeah, and one anti-fouling strategy that we thought of that's related to the conditions that the marine mussel needs is if all of these marine critters depend on using an acidic pH to keep their proteins in solution and to optimize conditions for binding to the surface, then engineering a surface that would maintain a high pH, even in acidic medium, might spell inability to attach for these organisms. It might be difficult, but there are a lot of polyamphiolites and polyelectrolytes and zwitterions out there now that could be tested. And you could add to that addition of surface topography to increase the density of those ionizable groups. This kind of adhesion, at least from the perspective of its cohesion, is like a thermoset, okay? You can't undo a thermoset because it gets cross-linked by these iron compounds, and those are still in principle reversible, but with dopa oxidation to quinone, you get covalent cross-links as well, and they won't be undone. But the interface, if it involves just catacols hydrogen bonded to the surface, assisted by the occasional cation, that should be reversible. And if I recall, in Messersmith's Nature 2007 paper, where he combined the Mussel adhesive with the gecko, he basically catechol functionalized a PDMS, I think, that was used in a gecko-like way to walk up a glass wall underwater. So that was reversible.
More question? Right. So, I'd like to thank Professor Waite.